Welcome to Longevity and Aging series. Today I would like to introduce Dr. Stephen Vatner. Dr. Vatner is a distinguished professor at Rutgers, New Jersey Medical School, serving in the Department of Cell Biology and Molecular Medicine. He also directs the Cardiovascular Research Institute at Rutgers University. Dr. Vatner's research focuses on cardiovascular control mechanisms in both normal and disease states. His work significantly advanced understanding of adrenergic neural control, exercise physiology, and myocardial ischemia. Dr. Vatner recently published a paper titled Brown Adipose Tissue Enhances Exercise Performance and Healthful Longevity. But before we start discussing the paper, Stephen, could you please tell us more about how you became interested in brown adipose tissue research?
We became interested
Yes.
with this model that
Mhm.
we developed. So another member of our department developed a model of longevity in fruit flies that related to RGS. And we looked up which one it would be closest to, and it would be closest to the regulator of GS protein 14. So we then developed a knockout of that model in mice. And that's when we found that its brown adipose tissue was unusually effective and accounted for much of its healthful aging.
Cool. Let's start then, maybe talking about adipose tissue. So, what are the main functions of white adipose tissue and brown adipose tissues? How are they different? And are there any detrimental effects, for example, of white adipose tissue? Because I think everyone talks a lot about detrimental effects, but are there any positive effects? And vice versa, are there any detrimental effect of brown adipose tissue?
Okay, well, most people know white adipose tissue has dual functions. One, it's sort of an energy reserve when needed to increase responses to acute stress. But then on the other hand, as you age, it has a detrimental effect, causing obesity, and the obesity is related to and causes diseases like heart failure, myocardial ischemia, cancer, exercise intolerance, and shortens longevity. Now, brown adipose tissue, on the other hand, doesn't have these adverse effects. And its main function is for thermogenesis and protect against the cold. But on the other hand, it has other functions that we and others have found out related to healthful longevity, increased exercise performance, reducing obesity.
And do we see any, what are the normal changes with age for these tissues? Obviously, we know that white adipose tissue increases with age. What happens with brown adipose tissue?
Oh, well, with age, it's a little controversial in the literature, but most people feel that brown adipose tissue declines with age. But there are some papers that have the reverse opinion.
And you discussing also that there are lots of research on effect of exercise on brown adipose tissue, right? So what are
Right.
these effects? How does exercise actually influence
Well,
brown?
exercise does increase brown adipose tissue. There's been a lot of articles on that, but relatively few articles on what we were interested in, how brown adipose tissue affects exercise. And we found that it has a very marked increase in exercise capacity, and that this RGS14 knockout mouse model has a much more profound effect than wild type brown adipose tissue.
Okay, so You said that you found this RGS14 gene interested because there were some effects in flies, right? Uh, and can you tell us more about the phenotypic effects of this RGS14 knockout in mice?
So they they increase lifespan, and interestingly, for mouse models, the females increase more than males, which is more like what's seen in humans. And as they increase their lifespan, they retain the characteristics of young mice. They don't have the characteristics of aging mice where they have atrophy and graying of hair, but we don't see that in the RGS14 knockout old mice.
Mhm.
Um, and they're also protected from obesity and many diseases.
Mhm. And what do you think are the mechanisms of this protection, of this increased longevity, basically?
Well, it it has many different effects. One, it um, it increases blood flow, so it and angiogenesis, which is very important as you age. It impairs it's antioxidative stress, has many antioxidative stress
Mhm.
mechanisms, which is bad for aging. Um, it improves mitochondrial biogenesis, which improves aging. So it has many mechanistic effects on improving aging
Mhm.
through UCP1 and SIRT3.
Where is it normally expressed, this RGS14 protein, and what is the function of this protein?
Which protein?
RGS14.
Well, it's a it's expressed in most tissues of the body,
Mhm.
and there are many RGS or regulator of GS proteins. This is just one of them. They all don't have the same effect. This one has more positive effects than negative effects.
Basically, uh, this protein, as I understand, it regulates uh, G protein alpha uh, GDP lifetime. And basically, when you have knockout, it means that protein MAP signaling should be activated, in my understanding. And this is probably uh, what determines to some extent the effects. So let's talk about the transplantation experiments. So I really like that in your paper you demonstrated that by transplanting brown adipose tissue, basically transplantation experiments basically show that the longevity effect of RGS14 is mediated through brown adipose tissue. Can you tell a little bit more about that?
Yeah, so what we did is surgically transplant the brown adipose tissue from the RGS14 knockout mice to their wild type controls.
Mhm.
Um, and we also transplanted brown adipose tissue from wild type mice to other wild type mice.
Mhm.
And what we found when we transplanted the brown adipose tissue from the RGS14 knockout to wild type is that the RGS14 knockout donor mice lost all their enhanced healthful longevity. And this was translated to the wild type mice with the RGS14 knockout bat.
Mhm.
They then experience healthful longevity, and when they get older, retain the appearance of young wild type mice. And then what's interesting, we compared in some cases transplanting the RGS14 knockout bat with transplanting bat from wild type to other wild type mice.
Mhm.
And we found that the bat from other wild type mice was not as powerful. For example, when we transferred the RGS14 knockout bat wild-type mice. The recipients, the wild-type bat recipients, increased their exercise capacity in 3 days. When we did this in wild-type to wild-type mice, the wild-type bat recipients took 2 months to increase their exercise capacity. So we believe that there's some unusual features of the RGS14 knockout bat
Mhm.
that's responsible for its healthful longevity.
Do you have any ideas of what exactly could be responsible for that, like more enhanced function of brown adipose tissue from knockout mice?
No, we don't. We're looking into that, but we haven't yet found out what
Mhm.
are the key differences between the bat from the RGS14 knockout and from wild type mice.
Mhm. But by the way, I'm curious, do you think, so I know that in the paper you showed that, and basically you studied longevity difference between wild type mice and knockout mice. But I'm curious if you had any experiments just to see the effect of transplantation on longevity. I mean,
Yes.
like if you just transplant, but does it actually increase lifespan in mice? I'm talking about even wild type mice.
Well, we do have data on transplanting the RGS14 bat
Mhm.
to uh, wild type mice and does increase their longevity.
Mhm.
But
Cool.
we don't know. We haven't studied the wild type bat to other wild type mice and its effects on longevity yet.
But I just didn't see this data in the paper. I mean, I didn't see the studies of longevity effects of bud transplantation. Uh, I only saw the survival curves between wild type and knockout mice. So I think it would be cool to see this kind of data if they're
Yeah, it's in a different paper, but
okay. Sorry. Um, it would be also cool to discuss the cellular mechanisms. So you showed in different paper that sirtuin-free is responsible for thermogenic effects of RGS14 knockout. Do you think sirtuin-free also could be responsible for this longevity effects?
yes, we believe so because we we developed a SIRT3 knockout mouse and mated it with the RGS14 knockout mouse.
Mhm.
And the RGS14 knockout mouse lost all of its healthful longevity in this dual knockout model. So the SIRT3 plays a critical role in mediating the healthful longevity and the positive effects on of bat in this mouse model.
Mhm. And what are the mechanisms of sirtuin-free effects? How does it actually affect the
Well,
longevity?
it stimulates UCP1 and which is the main thermogenic mechanism, and then it improves mitochondrial biogenesis. It also stimulates the increase in blood flow and angiogenesis, and through these many mechanisms, it increases exercise capacity and healthful longevity.
Mhm. Cool, very interesting. So, if you talk about uh, other mechanisms which are responsible for the brown adipose tissue effects, in particular the knockout mice, you also mentioned oxidative stress as a kind of decrease in
Yes.
oxidative stress as one of the mechanisms. Can you tell us a little bit more on that?
Well, the um, the bat and the RGS14 knockout through SIRT3 and UCP1 increase many uh, many mechanisms involved in protecting against oxidative stress. For example, MNSOD, but there are several others as well. And so this model is protected against oxidative stress, which is an important mechanism mediating healthful longevity.
And in what way is it protected? Like what kind of what kind of protection uh, does it show against what kind of stress? I mean, what kind of experiments demonstrate that?
It just, we haven't done too many specific experiments with the oxidative stress in this model, but it's well known that oxidative stress has many adverse effects
Mhm.
on healthful aging. And so it's generally believed that inhibiting oxidative stress would be beneficial, and that's what this model does.
Okay, so basically what we know at the moment is that some antioxidative enzymes like superoxide dismutase is overexpressed, and that's why we assume that oxidative stress should be lower just because it's important in aging process, right?
Well, there's several um, oxidative stress proteins that are increased in the RGS14 knockout model.
Mhm. Cool. Let's talk about potential clinical and therapeutic applications. So, do you know, um, like, I think wider audience would be interested in this. What are the lifestyle interventions potentially could be useful to improve gut activation or function?
Well, I think in order to do that, well, assuming that some of the same stresses improve BAT in humans as they do in the animal models, such as the increased exercise, exposure to cold temperature increases it. But I think the human studies really will depend on development of a pharmacological agent that mimics the RGS14 knockout bat. And we're working on that now, but that's going to take some time to develop and then show that it's safe enough to be delivered to humans.
Mhm. Sounds very exciting. And since you showed this exciting experiments on transplantation of brown adipose tissue, do you think the transplantation can be a strategy to improve health in patients somehow?
Yes, I think that the animal studies definitely have strong proof that it does, and that we hope that the same effects will be observed in humans when that's
Mhm.
time to do that.
How hard is it to do the surgery? Is it what are the side effects of that?
Well, obviously, for our type of surgery, you need a trained mouse surgeon, which we have, and it's not very difficult to transplant it because most of the brown fat in the mouse model, at least, is in the interscapular region in the back. And we then transplant it to the abdomen of another mouse. So it's relatively straightforward to do, and it's not a very dangerous surgery.
But where is it located in humans? And if you do this in humans, how dangerous could it be?
Well, again, I'm, I don't know where all the brown fat are located in humans. It's they may be in more diverse locations than in our animal models. And so that first has to be developed or found out. But I would expect that transplanting adipose tissue is never very dangerous as compared with working with hearts or pancreas or lungs surgically.
Okay, so this sounds quite exciting. Then, do you think also any RGS14 gene therapy could be used to improve health and longevity?
Yes, I believe that's going to be uh, another avenue of research and therapy. But I think it will first depend on development of the pharmacological analog
Mhm.
of RGS14 knockout bat. And then the gene therapy could be developed as well.
Mhm. And given that we develop this kind of therapies, for which diseases this kind of therapies would be most beneficial?
Well, it'll be most beneficial for any disease that has impaired exercise tolerance, which is a major impediment to healthful longevity.
Mhm.
It will have importance for any disease involving obesity, since the RGS14 knockout bat protects against obesity. And some of these diseases are heart failure, myocardial ischemia,
Mhm.
diabetes, um, even other diseases of the brain uh, are involved. So we have in our mouse models, we have evidence for all of these effects and being positive with the RGS14 knockout bat.
So, given this area is quite new and I think fast developing, what do you think are the most important challenges in brown adipose tissue research applied to longevity?
Well, I think the most significant challenge will be to translate these studies from animal models to humans. And I think the challenge there is how you can simulate RGS14 knockout bat in humans and increase its effects. And that's where we're back to developing a pharmacological analog, and the gene therapy fit in.
Mhm. Cool. Great. So yeah, very interesting paper, and I think yeah, and the approach is very promising as well. I would like to ask you also a general question about what area of anti-aging research do you think is, the most requires most extensive exploration? Well, obviously, maybe except the brown fat area.
Well, I think there's has to be a lot of research um, into developing healthful longevity in humans and translating. There are many models, maybe 40 models of healthful longevity in mice. And I think a lot of people are interested in translating some of these findings to humans, because even though lifespan, except for maybe the last year, lifespan is increased considerably over the past 50 years. Um, People are living longer, but they're not living longer healthfully, and they're suffering from many of these diseases of aging. So now that a lot of things have been found out to improve longevity, the next important step will be to improve healthful longevity and combat these diseases.
Great. Yeah. Hopefully, your upcoming research will help us to improve health span as well as longevity. Thank you very much for your time and participation in the podcast, and hope to see your new results soon.
Okay, thank you also.
All the best. Bye
Bye
bye.
bye.